Hello everyone, let's get started. Today we're going to be talking about three processes: endocytosis, exocytosis, and phagocytosis. Now the way we're going to approach these three processes is to first discuss the simplest one, which is exocytosis. We're going to talk about the mechanism of this process that helps to move materials out of the cell independent of any kind of concentration gradient. We'll also talk about the details of a special form of exocytosis called polarized secretion. Then we'll move on to the more complex topic of endocytosis, which is the process that moves materials into a cell, again, outside of any kind of concentration gradient. We'll discuss the mechanism of endocytosis. We'll talk about different forms of endocytosis, including phagocytosis and pinocytosis. And finally, we'll talk about the details of receptor-mediated endocytosis, the types of materials that come in through that form, how they are trafficked throughout the cell, the alternative processes involved in receptor-mediated endocytosis, and an important player in this process called coat protein. First, let's take a moment and review for ourselves how materials get moved around within a living cell, particularly things that travel through the endomembrane system. If you recall from earlier lectures, the endomembrane system is a system of interconnected membrane that stretches from the nuclear envelope all the way to the plasma membrane. The endomembrane system includes the nuclear envelope, the rough endoplasmic reticulum, transport vesicles, the Golgi apparatus, lysosomes, and the plasma membrane. All of this membrane is involved in the movement or the trafficking of particularly protein throughout the cell. Now, some proteins will reach the plasma membrane and have their fate be excretion from the cell or secretion from the cell. Again, this is a process that we term exocytosis, the purposeful secretion of materials out of a cell. In other words, independent of a concentration gradient. Similarly, when materials are fated to come into a cell across the plasma membrane and enter into the endomembrane system, they're going to enter in through a purposeful process called endocytosis. Endocytosis, exocytosis, these are processes that will remove materials or take materials in regardless of whether or not there is a concentration gradient. Now, let's look at some definitions for these terms that we're going to be talking about today. The term exocytosis refers to the expulsion or secretion of material from a cell by fusion of a transport vesicle with the plasma membrane. Endocytosis, in comparison, refers to the uptake of extracellular fluid and non-specific materials into a cell by infolding of the plasma membrane to form a transport vesicle. Now I'm going to point out that non-specific material is important here because there are generalized forms of endocytosis. There are also specialized forms of this process though, and we will talk about those. Those are the receptor-mediated type of endocytosis. Next, is this term phagocytosis. The word phago refers to eating, and cytosis or cyto means cell. So this is a form of cellular eating. And when we talk about phagocytosis, we're talking about materials entering the cell. So it is a form of endocytosis. Large particles or even entire organisms can be taken into a cell in order to be broken down or digested during phagocytosis. Now the key term here is large. We're talking about particles that are greater than about 0.5 micrometers, give or take a few nanometers. These are large pieces of material coming into the cell, or even entire organisms. Some other terminology that we need to be comfortable with are terms related to the small membrane-bound organelles that are involved in these processes: endocytosis, exocytosis, and phagocytosis. Remember, when we talk about a vesicle or a transport vesicle, we're using a very general term. We're describing a small organelle, so a membrane-bound compartment that is capable of carrying things around in a cell and merging with other membrane to move those materials into certain other organelles or even into or outside of a cell. The other terms we need to be familiar with surround the lysosome. Remember, the lysosome is very similar in appearance to a transport vesicle. It's a membrane-bound compartment, but it happens to have a very specific function. The lysosome contains acid hydrolase enzymes, which are capable of breaking down virtually all biologically relevant materials, with a few notable exceptions that we talked about. Now, it turns out that lysosomes do not just appear fully formed in a cell. They go through a maturation process. The term that we use to describe an immature lysosome is endosome. If you translate that word, it means the within body. So an endosome is simply a vesicle that is destined to mature and become a lysosome. We describe different stages of that maturation as being the early endosome and the late endosome. So an early endosome is a small organelle, a compartment again, like a vesicle, that emerges from the TGN. Now, I've not used that abbreviation before. TGN refers to the trans-Golgi network or the trans-Golgi apparatus, the trans side of the Golgi apparatus. Remember, that's the side that faces the membrane. The early endosome is mildly acidic in its internal compartment. Early endosomes can fuse with other vesicles. All vesicles can fuse with membrane, so early endosomes are no different from that. They can fuse with other vesicles, and they can continue to mature towards being a lysosome. Now, later in the maturation process, an early endosome becomes a late endosome. And we give them the designation of late endosome when they have become more acidic in their internal compartment. And I'm going to put some definitions on exactly what pH is what in just a few minutes. But for now, understand that early endosomes are mildly acidic in their interior, late endosomes are more acidic, and lysosomes have the highest acidity. Remember, the enzymes that do the function of molecular degradation and recycling in the lysosome are called acid hydrolases. And these enzymes require an acidic environment in order to become active. Let's take a look at this process known as exocytosis. Exocytosis is the final step in the pathway out of a cell for many secretory proteins. I have a list of some of the most common types of proteins that are routinely secreted from a cell. These include protein hormones, mucus, the proteins that are found in milk, and various digestive enzymes. All of these proteins are destined to function somewhere outside of the cell, and so must be secreted from the cell. Exocytosis sends vesicles to the membrane along microtubule tracts. Now, we haven't talked about the cytoskeleton yet, but for now, understand that one of the parts of the cytoskeleton is microtubules. And microtubules are literally linear filaments that can serve as roadways or railroad tracks, if you'd rather, inside the cell. There are specialized motor proteins that, as the name suggests, are capable of movement along these microtubule tracks, carrying these vesicles as they go. Now, we're going to be doing a laboratory exercise soon that involves cellular inhibitor chemicals, and one of those inhibitors is called colchicine. Colchicine is a toxin that inhibits the formation of microtubules. In other words, it prevents a cell from laying down the roadways that are required for exocytosis. Colchicine is one of the inhibitors that we will be looking at in our laboratory exercise. On this slide, you can see a diagram of the 4 steps of exocytosis. Let's walk through them together. In the diagram, the light orange represents the cytoplasm inside the cell, and this structure represents the membrane, the plasma membrane. The blue area represents the exterior of the cell, and the diagram is meant to be read from the top to the bottom. Now, you'll notice that there are these strange markings along the plasma membrane. Those represent proteins. Remember, living cells have plasma membranes that are loaded with proteins. So let's start with number 1 at the top of the diagram. What you're looking at here is a representation of a transport vesicle. You can see it's surrounded by a membrane, and inside it contains all of these little black dots that represent some kind of a protein that is destined to be secreted from the cell. Now, the first thing that has to happen is this vesicle has to come up to approaching the membrane. Now, the vesicle and the membrane have to come together and fuse. The protein that is sitting in the membrane of the vesicle has to interact with and bind to protein that is in the plasma membrane in order for this fusion to occur. The third step in the process involves the rupture of the membrane. So there will be a physical opening created where those 2 membranes have fused together. And finally, all of the contents inside the vesicle will be discharged to the outside of the cell, as they're showing here. Now you might wonder what happens to this empty transport vesicle at this point. And the answer is it simply becomes part of the plasma membrane. In other words, its membrane simply melds in and becomes part of the plasma membrane. Exocytosis is not a complex process on its face, although there are some additional players involved. The fusion of the vesicle, for example, with that plasma membrane is triggered by an event. And that event is brought about by what are called second messengers. Now, when we use this terminology in cell biology, we're talking about cascades of signaling that happen within cells. As you know, compounds commonly interact with and bind to protein receptors on the surface of a membrane surrounding a cell. Molecules can come in and bind to protein receptors. Remember, anytime something binds to a protein, that protein is going to change shape and therefore change function. So when molecules bind to protein receptors, the shape change that occurs will cause something else to occur in the cell. This is what we call a cascade. And sometimes these cascades can continue on. The event that gets triggered in the cell will then cause another event to occur in the cell, and so on and so on. In the case of exocytosis, it's molecules like hormones and neurotransmitters that are coming in and binding to receptors on the membrane. You might consider these the first messengers. That binding event of the hormone or the neurotransmitter to its receptor on the membrane is going to cause that protein receptor to change shape. And that shape change is going to activate a second messenger inside the cell. Now, in the case of exocytosis, this second messenger is calcium. In fact, calcium is a very common second messenger in lots of processes in living cells. So when the first messenger binds to a receptor, it activates an increase in intracellular calcium levels, and particularly calcium ions. This increased level of calcium ion is going to activate a type of enzyme called a protein kinase. Now, kinases are a fascinating family of enzymes. They are a huge family, what we call a super family of enzymes, and they have one job. That job is to phosphorylate other proteins. In other words, kinase enzymes function by placing phosphate groups onto proteins. And when they place these phosphate groups onto the protein changes shape and changes function. So, in order for exocytosis to be initiated, some hormone or neurotransmitter or other type of molecule has to bind to its receptor on the surface of the cell, telling the cell it's time to secrete this protein. That binding at the surface of the cell triggers an increase in intracellular calcium levels. The high calcium then activates protein kinase enzymes. And those protein kinase enzymes then phosphorylate protein, specifically the proteins that are on the surface of transport vesicles that are containing this protein that needs to be secreted. The protein kinase enzymes also phosphorylate proteins on the inside of the plasma membrane. Now we have proteins on the outside of the transport vesicle and the inside of the plasma membrane phosphorylated. That's what allows them to come together and fuse. Remember, those two membranes have to fuse with each other in order to then rupture and release the secretory proteins. It's the phosphorylation of the outside of the vesicle membrane and the inside of the plasma membrane that allows those proteins to come together and fuse so that the material inside the transport vesicle can be released or secreted to the outside of the cell.